Assalamu alaikum everyone. I'm Ikra and my instructor is Professor Sadaf Javed. So I'm gonna talk about the pre-process of utilization of fatty acids. Not about the utilization of fatty acid, but there is a very important pathway which leads this utilization of fatty acids. And this pathway is called Carnitine Shuttle pathway. So what is carnitine? It's a ammonium compound and it plays a vital role in transport of fatty acid from cytosol to mitochondrial matrix for the generation of energy. So here is the structure of carnitine. In the next slide, let's see how and where it synthesizes. Carnitine basically synthesized in liver and kidney. Moreover, it synthesized by two amino acids, which is lysine and methionine. Actually, small and medium-sized fatty acid can easily pass through the mitochondrial membrane, but the long-chain fatty acids, about more than 40 carbons, needs carnitine to shuttle inside the mitochondria. Here we'll talk about why and when this process occurs. Actually, if a person is starving and fasting, especially those who do keto diet, there is a low level of glucose for energy production. So our bodies start to generate energy from fatty acids. As you already know, fatty acids stored in adipose cells. These fatty acids transport to the blood, and in the blood, fatty acid will bind to the albumin, which carries fatty acid into the target cell. So before its transportation into the mitochondria, fatty acid need to be activated. So how it will activate? Fatty acid will bind to the coenzyme A, and which is already present in the cytosol of cytoplasm, and this process need ATP in the form of AMP, adenomonophosphate, which drives a thioester linkage between coenzyme A and fatty acid A. After binding, an acyl-CoA will be formed, and now this acyl-CoA is ready to transport, which is carried out by an carnitine. Carnitine is acting as a carrier, or you can say a loading compound. So acyl-CoA will bind to the carnitine, and this process occurs in the presence of an enzyme which is called Acyltransferase 1. I'm repeating, Acyltransferase 1. You have to remember this enzyme, which is located outside the mitochondrial membrane. Let's see this whole process with a diagram for your better understanding. So here is the diagram. On the left side of the diagram, you can see the reaction in purple color. This is all about the activation of fatty acids. We have already discussed about this. So acyl-CoA will bind to the carnitine to form acyl-carnitine. Actually, carnitine will shift at the place of coenzyme A. Coenzyme will be replaced with carnitine, and an acyl-carnitine will be formed, and this whole process is carried out by an enzyme is acyltransferase 1. You can see the yellow bean shaped is showing the acyltransferase 1. Now acyl-carnitine is ready to shuttle inside through a carrier protein. The blue box shows the carrier of transport protein, which is located inner mitochondrial membrane. Now, finally, acyl-carnitine reached inside the mitochondrial matrix, and again there gonna be a reverse process. The carnitine of acyl-carnitine again exchanged by coenzyme A, which is present inside the mitochondrial matrix. And that's how carnitine will be separate from the acylcarnitine and came back out of the mitochondria through a transporter protein to carry and other fatty acids. After reaching inside the mitochondria, fatty acid will undergo beta oxidation to form acyl-CoA, and which will further undergo for the Krebs cycle to generate energy. So that was all the whole reaction. Now let's see its regulation. And there is another process, yeah, in the cytosol there is another process, the formation of fatty acids. In this process an intermediate compound formed which is called malonyl CoA, and this malonyl CoA inhibit the enzyme Acyltransferase 1. Do you remember this enzyme? I have told you to remember that. So what I'm trying to say, that if one process is in progress, other process will be stopped. These two process, the formation and utilization of fatty acid, can't be occur simultaneously. If the process formation of fatty acid is happening, the utilization of fatty acid means carnitine all process will be stopped. So what if our body is deficient from transporter carnitine? What will happen? Those people who have carnitine transporter deviation might not be able to use their fats as energy production. And what will be the symptoms? Muscle weakness, aching, and muscle cramps. So that was all about the Carnitine Shuttle system. I hope you all have get that, and thanks for watching this video. Allah Hafiz.